Hello, everyone, and welcome to today's webinar. I'm Melissa Romero, Forensic Science Consultant and CNN Media Group. I will be moderating today's event, Integrated Screening and Assay Compatibility Facilitate Peptide Drug Early Discovery. This educational webinar is sponsored by WuXi AppTec. CNN works with sponsors to identify topics of interest and value to CNEN's audience and consistent with CNEN's mission to provide news and analysis of the chemistry enterprise in a timely, accurate, and balanced fashion. During the webinar, you can adjust the size of the slides on your screen by grabbing the lower right corner with your mouse. If you need technical assistance, please look at the Help widget at the top right corner of the screen or type your query into the Q&A box. If you are disconnected during the webcast, please log in again according to the instructions you received earlier. You're encouraged to contribute to the success of this webinar by asking questions at any time during the presentation through the Q&A box on your screen. The questions will be answered at the end of the presentation, allowing for as many as time permits. This webcast allows certification of participation. To receive your certificate, please click on the certificate icon below your Q&A box to download your copy after watching the webinar. Please note that CNN does not endorse any company, products, or services that may be mentioned in the webinars, and each webinar will be archived at CNN Online after the live webcast. As mentioned, this educational webinar is sponsored by WuXi AppTec. As a global company with operations across Asia, Europe, and North America, WuXi AppTec provides a broad portfolio of R&D and manufacturing services that enable the global pharmaceutical and life sciences industry to advance discoveries and deliver groundbreaking treatments to patients. Through its unique business models, WuXi AppTec's integrated end-to-end services include biology discovery, preclinical testing, and clinical research services, and chemistry drug CRDMO, which is contract research, development, and manufacturing organization. These help customers improve the productivity of advancing healthcare products through cost-effective and efficient solutions. WuXi AppTec received an AA ESG rating from MSCI for the fourth consecutive year in 2024, and its open access platform is enabling around 6,000 customers from over 30 countries to improve the health of those in need and to realize the vision that every drug can be made and every disease can be treated. During the presentation today, we will be hearing from Dr. Zhengyao Mu and Dr. Yifei Singh. Dr. Zhengyao Mu is Director WuXi Biology at WuXi AppTec and holds a bachelor's degree in biology from Shanghai Jiao Tong University and a PhD in genetics and developmental biology from the University of Georgia in the U.S. His research focused on thymic epithelial cell reprogramming and developmental regulation. In early 2021, Dr. Mu joined WuXi AppTec and assumed a position at the China Research and Development Center. He is responsible for expanding the application of DNA-encoded library, DEL technology, and developing screening models for DEL in new modality compounds such as peptide and molecular glue, as well as new target areas. Dr. Yifei Singh is Director of WuXi Biology at WuXi AppTec. Dr. Singh was trained as a biochemist with a bachelor's degree from Nanjing University and received her PhD in physiology from the Medical College of Georgia, majoring in molecular and cell biology. She joined WuXi AppTec in 2024 and serves as the Director of Peptide Platform Hit Validation, focusing on offering comprehensive early-stage peptide drug discovery services for clients and continuously developing integrated solutions to address novel modality discoveries. Now let's get started. I will turn it over to Dr. Zhengyao Mu.
Thanks for the great introduction, and it's our pleasure to have these opportunities to present our WuXi integrated peptide discovery platform. So today we're going to show our overall capabilities of how we help to discover the peptide hit and how we help our clients to validate these peptide primary hit, and then how we actually apply the peptide hit into actual application scenarios. So first of all, please let me to briefly introduce why peptide is so important, why currently peptide market is so hot, and why WuXi want to build up our integrated peptide discovery platform. So as we can see in the figure, peptide lies right in the middle of the small moleculars and proteins. So considering the molecular weight, the peptide has a bigger molecular weight compared to small moleculars, which give it unique advantages. For example, high target specificities, high target affinities, fewer side effects, and also less accumulations in tissues. And in the other side, if we compare peptide to traditional therapeutic proteins or therapeutic biologics such as antibodies, the peptide has lower molecular weights, which also give it unique advantages such as better membrane permeabilities, less immunogenic, and also the synthesis of peptide is way easier compared to traditional therapeutic proteins and biologic antibodies. So overall, these advantages give peptide very good, unique, competitive, such as highly potent and selective, and also less toxic. That's why more and more people are focusing on peptide drug discoveries. So peptide drug actually have several different versions. For example, like linear peptide, cyclic peptide, and also conjugated peptide. In the other way, radioisotope labeled peptide is also constantly growing. Starting from 1940, if we take a look on the history of the peptide drug discoveries, we can see there's an increasing trend of the peptide drug getting approved by the FDA over years. So for example, in the 2010 to 2019, there's about 33 different peptide drugs getting approved by the FDA. And only within the 2020 to 2022, these 3 years, there's already 13 different peptide drugs getting approved by the FDA, and the number is still growing. So peptide has been shown and approved to have the abilities to cure different kind of diseases, and more effort of the clinical research has been put into the peptide drug. If we take a look on the current peptide drug market, the most popular peptide drug must be the semaglutide, which its target was actually the GLP-1. So if we take a look on the figure on the top right, we can see the semaglutide can significantly decrease the body weight over time comparing to the placebo, which is make it the most popular drug in the market in 2025. But actually, the diabetes and the body weight loss is not the only field that peptide can be focusing on. If we take a look on the bottom right chart, we can see actually there's a lot of clinical trials ongoing, and they are focusing on different kinds of scenarios, such as infections, diagnostic agents, and oncology potential therapeutic application scenarios. So peptide drug has wide field of application disease, and we can actually use peptide drug to cure different kinds of potential disease serotype. So peptide normally have linear peptide and cyclic peptide, and actually peptide can be combined with other modalities to further expand its potential therapeutic applications. For example, on the right side, we can see peptide can be conjugated with either small molecular drug or toxins to make it into a PDC for further applications. And also, peptide can also be conjugated with the oligonucleotide so that it could be used to help deliver the oligonucleotide drugs such as siRNA or ASO to its disease type cellular or disease-related organs. And another thing that peptide can be used is actually radioisotope labeling peptide. We can directly label those high high binding affinity peptide with a radioisotope so that it can be used in actual disease curing. So in WuXi, we actually provide end-to-end integrated services for the peptide drug discoveries. We actually have three major segments: WuXi biology, WuXi chemistry, and the WuXi testing, all covering different phases and steps of the peptide drug discovery. For example, for the WuXi biology, we actually provide services for target identifications, protein science, hit discovery, in vitro and in vivo primary hit validations, and the follow-up animal models. For the WuXi chemistry, we help our clients to synthesize these complicated peptide primary hits, and also we offer medicinal chemistry services to further optimize these structures. And after the first synthesis of the peptide drugs, we can even scale up of the peptide synthesis to help our clients to further move into further animal studies or even clinical models. For the WuXi testings, it is very important for the peptide drug compound profilings. So WuXi testing can actually help to test the peptide drug permeabilities, serum stabilities, the in vitro ADME, the in vivo PK, and also the most important thing, the safety assessments. So coming back to the primary peptide drug discovery process, in the WuXi biology, we actually offer three different kinds of peptide drug discovery solutions. On the left part, it was peptide DNA-encoded libraries. Basically, it was highly similar to traditional small molecular DNA-encoded libraries. We built up these peptide libraries using split and pool combinatory chemistries. So the overall diversity of this library is pretty good. We can actually reach to 10 to the 11 diversities in this peptide drug discovery libraries. So the most special thing that we have to keep in mind of this peptide drug discovery library is that actually we use a huge amount of unnatural amino acids into these libraries. Actually, for our off-the-shelf libraries, we have more than 500 different unnatural amino acids. And also another unique advantage of these peptide libraries is that we can utilize a lot of different cyclization reactions. Comparing to traditional display technologies, which mainly focusing on cysteine-cysteine disulfide cyclizations, peptide DNA-encoded libraries give you more opportunities and choice of the monocyclic peptide cyclizations. On the right side, we can see this is the very popular display technologies. So we can mainly put them into two different kinds. The first one is actually the phage display, and the second one is the mRNA display. For the mRNA display, the most unique advantage is the library diversities. As we can see on the figure, the overall diversity of the mRNA display can actually reach to 10 to the 12, and you can even further increase the in vitro translation system to get a higher diversities. Sometimes it can reach even to the 10 to the 13. And another unique advantage of mRNA display and the phage display is that actually you can design your unique library structures by directly synthesizing those DNAs, and is synthesizing oligos is quick and easy. You can build up your own peptide library very quickly and cost efficiently. For the phage display, although the overall diversity is a little bit lower, which is normally 10 to the 9, it also has its unique advantages. So after the primary screenings, we can use single phage ELISA to filter those primary signals so that we can pick up those hits with some higher confidence. So today I'm gonna bring up two different stories. of how WuXi use these peptide drug discovery technologies to facilitate the overall peptide primary hit identifications. So the first one, the story I'm gonna talk about, is peptide DNA-encoded library facilitate novel potent MDM2 p53 microcyclic peptide inhibitor. So as we discussed before, we have a lot of different structures of the DNA-encoded library peptide libraries. So just like what we show on the slides, we have both, linear peptide and also cyclic version of peptides. All of these libraries have its unique advantages. For example, for the diversities, we have more around 100 billion diversities for the linear peptide libraries, and for the cyclic peptide libraries, the total diversity reaches about like 150 billion diversities. And also on the right side, when we design our off-the-shelf libraries, we actually carefully design and monitor the building blocks we pick of these libraries. For example, we choose both D and L amino acids to mix them in a pretty equal ratios, which is 3 to 4. And also for the modifications, methyl modifications and alkylations is pretty important for the physical properties of these peptide drugs, which may help them have higher stabilities in the serum and also in further animal studies, and also give them better permeabilities. That's why we actually take about 10% of our library building blocks to be these modifications. So the ring size of the cyclic peptide is also pretty important. So in our off-the-shelf libraries, the ring size is actually 6 to 9 amino acids, but we are keep expanding the size of the peptide libraries to about 12 amino acids. As we talked before, one of the unique advantages of the peptide DNA-encoded libraries is we can using a lot of different versions of the cyclizations. So for example, like the click reactions and other cyclization methodologies, as well as the linkers. So another hot topic in the peptide drug discovery field is about oral peptide. One solution to help the peptide to be more oral bioavailable is to decrease the peptide size, but in the same time try to maintain its high potency. So we should try to achieve that goal by making small cyclic peptide libraries with different natural amino acids. As we can see, we have two off-the-shelf libraries. The first one is about like 16 million diversity, and the second one is about like 51 billion diversities. And we're using around 22 different linkers for the cyclizations to further increase the chemical space and diversities. So we believe these physical chemical properties, these small molecular weight, these better log-P design, and these different linker cyclization strategies can help to find small cyclic peptide, which might be more oral bioavailable. So now people are moving into more and more cyclic peptide because cyclic peptide has its unique advantages comparing to linear peptide. For example, cyclic peptide has better oral bioavailabilities, and also it has better resistance to the peptides. But the most important thing of the cyclic peptide is actually its conformation is more constrained and stable, which make it to be binding more specific to its potential target, which can further decrease the off-target effects. So using DNA-encoded library to building up a cyclic peptide library to screening for potential peptide drug is one of the key things that we want to prove for the past several years. So actually, in this case studies, we would like to show how we're using our cyclic peptide DNA-encoded libraries to identify the MDM2 p53 inhibitors. But before we actually go into the stories, please allow me to basically introduce how the DNA-encoded library looks like and how the DNA-encoded library screening gonna looks like. So as we can see on the left top, we can see the actual DNA-encoded library is contains two major parts. The first part was actually the warhead, which is actually the chemical structures and the chemical compounds. In our case, it's gonna be our cyclic peptide. And on the right part is actually different DNA tags, which encodes the corresponding peptide amino acids or building blocks. So through these split and fork construction strategies, we can actually identify the post-selection peptide structures by directly sequencing the DNA barcodes. So once we construct the DNA encoded library, then we're going to move into the actual affinity-based selections. So the affinity-based selection for DNA encoded library is pretty straightforward. First of all, you need a very high quality protein. You immobilize the protein into the affinity matrix, and then you're going to incubate your DNA encoded libraries with the proteins. After several rounds of the wash, then we elute the binder stay in the proteins. After several rounds of the binding and elute, we can then use the NGS to identify the barcodes to further understand what peptide or small molecule are actually binding to the target protein. And then we can using off-DNA resynthesize to confirm whether these hits are real or not. So one of the major advantage and beauty of the DNA encoded library is that it requires minimum assay development so that most of the target, as long as the protein is available, we can use in DNA encoded library to screening for the target. And another thing is that DNA encoded library have a huge amount of the diversity. And since we are using split and pour combinatory chemistry to building up this library, we can also get a huge amount of SAI information directly from the screenings. And on the right side, this is an excellent example of how in the DNA encoded library we can see some primary SAI informations. As we can see on the right cube plot, we see several blue dots lined straight up. This indicates these peptides after the selections share very high similar structures, and these structures might be super important for the target compound binding MOA. So after we identify these SAR, we can further identify the most important part of the protein compound binding. So after we're building up this DNA encoded library, and then we're gonna move into the actual screenings to make sure we can use cyclic peptide libraries to identify potent peptide drug. So we actually do this POC with one of our excellent collaborators, and the final result was published in 2023. So in this case studies, we first built up in 4.5 billion member cyclic peptide DNA encoded libraries with its unique design strategies. For example, we want a lower molecular weight. We also want high backbone diversities. And also we want the structure looks a little bit different comparing to phage and mRNA displays. And after construct such unique cyclic peptide DNA encoded libraries, we perform the screenings with our collaborators against the MDM2 and the p53, and luckily we directly identify a very potent initial primary hit, which shows single-digit nanomole IC50s in the HTIF assays. Another unique advantage of the peptide DNA encoded library primary hit identifications is that we can combine different cyclization methodology sublibraries together and directly compare their performance in the screenings. And this is actually one of the most exciting part that I find when we actually analyzing the data. So this for in the figure we can see on the slides, we can see there's 3 different compounds. They all share exactly the same building blocks, 401, 415, 375, and 180. But the only difference of these 3 different compounds is actually the initial library ID, which is 577, 578, and 579, which is indicating they have different cyclization strategies. So if we take a look on the post-selection performance, we can see only one unique cyclization strategy shows its enrichment post after the selections, and the other 2 didn't really have any enrichment signals. This tells us that this unique cyclization strategy is really important for this compound to binding to the target proteins. So after we off DNA synthesize these compounds, we can see this compound has very high IC50s, which is mainly single-digit or 2-digit nanomole binding affinities in the IC50s in the HTF assays or as well as in the FP assays. And the further structure analysis also shows these primary hits binding to the MDM2 in a very its binding mode to its target protein MDM2. So to summary, for the peptide DNA-encoded library part, we have experienced DNA-encoded library construction experience, and then we further utilize these previous experience into the peptide DNA-encoded library constructions. A large amount of unnatural amino acids is one of the key strengths for the peptide DNA-encoded library, and also the library design, the library construction is highly customizable. In our case studies, we show that potent MDM2p53 inhibitor is successfully identified from the peptide DNA-encoded libraries with desired properties. And finally, the direct comparison of linear and cyclic sublibraries is one of the key strengths for peptide DNA-encoded library. For the second story, I would like to basically talk about how we're using mRNA displays to looking for the primary hits and how mRNA display generates sufficient cluster informations in the post-selection NGS analysis. So before we run into the actual case studies, let's take a look on how the mRNA display workflow gonna looks like. So normally an mRNA display gonna starting with a double-strand DNA oligo synthesize. Based on what kind of peptide structure you would like to have in your screenings, you're gonna synthesize the corresponding DNA oligos. And then we're gonna do the in vitro transcriptions to make the DNA into RNA. And remember, on this step, the diversity of the library is already determined. So in most cases, the mRNA library is pretty high, around 10 to the 12 diversities. And once we got the mRNA, then the most critical step of the mRNA display gonna be the pyrimidine linker ligations. We're gonna ligate the set of linkers which conjugate with the pyrimidine to the mRNA, so that we can make sure the mRNA gonna stay with the peptide in the following selections, so that we can further PCR or NGS the post-selection samples. So once we finish the pyrimidine ligations, we're gonna further move into the in vitro translations to translate these mRNA into corresponding peptide. So these peptides are mainly still linear versions. So then we have to do the cyclizations based on the actual design of the library. Sometimes you can directly using the cysteine disulfide cyclization. Then sometimes you can pick the small molecular linker to make the cyclization happens. Different to the DNA-encoded library screenings, in which we're using parallel selections, in mRNA display-based selections, we're gonna first do a negative counter selections, in which we can using the affinity matrix or the counter proteins to pre-clean the mRNA display libraries. After the pre-clean step, we're gonna take the flow through into the actual binding selections, in which we will immobilize the target protein to the affinity matrix, and then we incubate our mRNA display libraries with target proteins. After we elute the libraries from target proteins, then we can further PCR these elutions to construct the oligo libraries for the second round. So based on our previous experience, the mRNA display library selections normally takes 5 to 10 rounds until we see a clear signal of enrichment. So how we can determine that the selection is going into the right directions? The first thing and the most straightforward way we can do that is actually qPCR-based analysis. So as we can see on the right figure, we can see on the first several rounds, the post-selection enrichment is actually lower or comparable to the negative selection. But starting from the fourth round, we see a significant difference between the positive selection enrichment and the negative selection enrichment in the qPCR analysis. And then further goes into the fifth round, we can even see the positive selection is way higher than the negative selection. So this tells us the selection is going well. And after we take the fifth round selection samples and send them to the NGS, we can see in the NGS data, clear sequence clustering is observed, which means we identify a huge amount of peptides which share a very high sequence similarities. And this tells us these sequence probably is the right one. They're getting enriched through rounds to rounds. So let's move into a real case, which is actually a cell surface receptors. So in this cell surface receptor selections, and this is actually a pretty challenging one, we would say. So we actually, in the first fifth round, we don't actually see a very significant difference of the positive samples compared to negative samples. But after the sixth round, we see a significant difference of the positive samples and negative samples. And actually the positive samples reached the plateau at the eighth round, and the negative samples actually goes to a very low amount. So we send out the final rounds of the samples to NGS to see whether we can see some potential clusterings. As we can see on the right, for the positive samples NGS data, we can see a lot of different cluster blocks, which is showing as a blue dark box. This tells us that we do identify a lot of peptides with high sequence similarities. However, when we take a look on the negative selection samples in NGS, these peptide sequences are mainly random sequence. They don't have a huge sequence similarities, which tell us they're just random background noise. So for cell surface receptors, one of the important thing is that sometimes we don't want binding to its orthosteric pocket, because orthosteric pocket may binding to its natural protein binding partner or natural ligand in the in vivo environment, so that even you identify very high potent orthosteric binders in the primary selections, it may not have remain its potencies when we further evolve into animal studies. So in order to identify an allosteric binders, you have to develop your unique selection strategies. So in our cases, we include in special round 7 selections so that we can identify both allosteric binders and orthosteric binders. For the allosteric binder selections, we actually pre-block the pocket using a very high potent natural binding ligand, and then we incubate our libraries with our target protein so that we can get some allosteric binders through these competition-based blocking strategies. So after we sending out the round 7 samples into the NGS, one interesting thing that we observe is that although we mix libraries lengths from 9 amino acids all the way to 14 amino acids gathered in the selections, there's only 12 amino acids, 13 amino acids, and 14 amino acids showing up positive enrichment and also clusterings after the NGS, indicating these lengths is probably more favorable for this cell surface receptor selection in the pocket. So whether our selection strategy works, we actually often synthesize both potential allosteric binders and also potential allosteric binders and validate them using SPR assay. As we can see on the left, for the allosteric binders, it shows very strong binding affinities, which is 2-digit nanomole in the SPR assays to the target proteins. But when we actually put into the nature ligand to do the computations, there's no binding observed at all. For the allosteric binders, actually, it also shows a very strong binding to its target protein when it's present alone, which is single-digit nanomole binding affinities in the SPR assays. But when we actually putting in the natural binding ligand to the target protein, this allosteric binder peptide still maintains a very strong binding affinities, which is around 2-digit nanomole, which indicating our allosteric binder selection strategy is working. So for this part, the take-home message is that multiple rounds of negative and positive selection was conducted for protein of interest using nature amino acid-based mRNA display. And in our POC target selections, obvious peptide sequence clustering was observed by post-selection NGS analysis. After peptide hit resynthesized, 2 peptides shows nanomole binding affinities against the target proteins, while one of them is potential allosteric binders. But identifying the primary peptide hit is only the very, very initial step. After we find the primary hit, there's still a lot more work needs to do, and that's why we need an integrated bioassay platform for the peptide profiling. And our colleague Ye will cover and introduce how WuXi do the peptide profilings using our integrated bioassay platform.
Thank Zhongyao for the nice introduction on the screening capability. So after screening, after we get a potential list of potential hits, the natural question next is, are those real binders we're looking for? What are those binders? Do they have the function we're looking for? And do they have the right characteristics we're looking for as a drug? So all those questions need to be answered in the sequential characterization. So based on those needs, we have built the integrated bioassay platform for peptide profiling. As you can see here below, we actually focused on 5 key features people are interested in peptide drug discovery field. So the first one is actually its binding affinity to the target, whether it can disrupt the protein-protein interaction we're looking for. And the second one is actually permeability, especially if the target is intracellular target, can the peptide penetrate into the cell to bind to its target to do its function? And also if it can penetrate the GI tract to be successfully absorbed into the circulation, and also if the target is in the CNS, can the peptide penetrate into the BBB barrier? So all those need to be characterized. Then the third thing we are interested in is actually the stability of the peptide, whether in the circulation or in the tissue inside the cell, would it be stable enough to be able to reach the target to effective dose. Then the fourth one, of course, is functionality, whether it can do what it's designed to do at the area of target. Then the last thing is specificity and toxicity. And then based for those 5 key features, we have designed a series of different assays, including biophysical assay, biochemical assay, structural biology, cell biology, and ex vivo models to be able to fully characterize those 5 key features. So altogether, we build a comprehensive toolbox for peptide profiling. But as you can see, this is like a big list. And then people will ask, how do I choose the right assay to characterize the peptide I have found? So that's case by case. It depends on the target you're looking for. depends on the disease applications. But in general, we have two recommended workflow that are designed for different applications. First, if the peptide is designed as a therapeutic drug, which means it's function directly to the target to exert its treatment effect, then we would recommend to start with a biochemical and a biophysical assay to characterize its binding and protein-protein interaction disruption function. So we would recommend either ELISA or TR-FRET and SPR to be able to find how good the peptide is a binder to the target. Then secondly, we would recommend to move to cellular assay to characterize its function and potency. This we can use the peptide either on a reporter line or the primary cell models based on the disease application. After that, if we find the peptide can be a good binder and it has the designated function we're looking for, the third thing we will look at is actually its metabolite ID, which need to be found like how stable it is, for example, in the serum or plasma. What are the soft spots that can be digested in the blood or by different enzymes? So combining all those informations, then we can go through several rounds of peptide optimization to further improve the performance of peptides. After that, we will get a pretty potent and promising PCC that we can move into the full profiling package to look at its permeability, its functionality, and also the in vitro toxicity and immunogenicity of the peptide we found. And lastly, we will move to animal models to look at the in vivo DMPK and also using different disease models to look at its efficacy. So all the process, depending on the rounds of peptide optimization and the performance of the peptide, it can take several months to years to go through the whole process. However, if we're looking at the application of using peptide as a delivery tool to deliver cargoes into the body, the process can be a little simpler because in general, we're just looking at the delivery efficiency of the peptide. So for this application, we would still recommend to look using ELISA, TR-FRET, or SPR to first look at how good the peptide binds to the target. But then secondly, we would quickly move into a cell surface binding and internalization capability characterization, either using flow cytometry or confocal microscopy methods. Then a quick stability test in the plasma or serum and in the hepatocyte will give us information for the peptide optimization. After that, we will recommend doing conjugation manufacturing and then do the full in vivo DMPK and toxicity test of the peptide with the cargo on it. And this process can be a little shorter depending on the performance of the peptide. So those are the two general workflow we have designed for peptide profiling. And now I'm going to use two showcases to demonstrate in real life how do we use those workflow and the toolbox we build to help facilitate the drug validation and the improvement. So the first showcase I'm going to demonstrate is actually using the peptide for therapeutic application. And the target using here is actually PCSK9. I guess most of us are pretty familiar with this target since Merck's MK0616 has shown very promising phase 3 results recently. So the PCSK9 has becoming a pretty hot target in the field. And the mechanism for PCSK9 is actually, it is hepatocyte secreted protein. So when it's existing in high level, it actually can bind to the LDL receptor on the surface of the liver. And then when it binds to the LDL receptor, it actually facilitates the endocytosis of the receptor and then drag it into the endosome for degradation. So with a high level of PCSK9 present, actually it will deplete the cell surface LDL receptor, which lead to decreased LDL metabolism in the liver and leads to high levels of cholesterol in circulation. So for this target, the drug discovery rationale is actually to find a blocker that can block the binding of PCSK9 to LDL receptor. That way, the cell surface LDL receptor levels are maintained to be able to clean the LDL in circulation. So with that in mind, we have set up our screening campaign using the Peptide-DEL platform. So for this one, we actually set up two conditions. One is to screen the library with PCSK9, the target protein only. And the second one is actually adding the MK0616, which is the antagonist to PCSK9 to block the binding pocket. So with those two conditions set up, and after the screening, when we compare the two sets of binders found in those two conditions, we actually can find the population one, which is showing in blue here. Those binders are actually the peptides that can bind into the pocket for PCSK9, while the others show in green actually are allostatic binders for PCSK9. So after that, we do see enrichment of a number of peptides that are enriched in different conditions, and also they are different in structure. Some of those are cyclic peptide, some are linear peptide, and they have different sizes as shown here in the middle. From those data, after the post-selection data analysis, we actually identified a group of peptide we think are promising with high enrichment and high potential as a binder. And after that, we did a whole full package of the peptide profiling using a triaging funnel shown here. So out of the 250 billion peptide existed in the Peptide-DEL screening library, we picked 38 of them for resynthesize. And out of the 38 that resynthesized, we run off-DNA ASMS to look at binders, and 22 of those actually have pretty good performance. Out of those 22, we also did another biophysical assay, SPR, to look at the KD of this peptide, and 18 of those actually have the nanomole range KDs from the SPR. And then out of those, we also did some biochemical assays, including ELISA and TR-FRET. And we have identified 8 of the peptide actually confirm the good binding with a biochemical assay. And we are currently confirming the function of those 8 peptides inside the cell to make sure those are the real blockers for PCSK9 binding to LDL receptor. And then we also look at the metabolite stability of those compounds we have identified. As you can see here, one of the beauty of the DEL screening actually is because we have included so many unnatural amino acids in the library. So the peptide we find actually are having a pretty good stability in serum. As you can see here on the right, in the human serum, most of the peptide we tested actually have more than 60 hours of half-life, making them a pretty good bioavailability for drug development. So in summary, we have used the DAO technology, which can be a pretty useful tool for peptide discovery. And because of its high component of unnatural amino acid in the library, the peptide hits we find from DAO screening normally have advantages in stability and affinity. And also we have shown that we successfully using the triaging profiling workflow combining biophysical, biochemical, and cell-based assay can enable the finding of quality hits from screening result and can increase the success rate of PCC finding. And as shown here, advanced peptide modalities such as cyclic peptide can enable normal therapeutic application. So the second case I'm going to show is actually related to use peptide as a delivery tool. So peptide actually has been gaining more and more interest to as a delivery tool because it's compared to the small molecule. It has better affinity and better specificity, and also because compared to the antibody ADC, antibody-related drug, it actually has less potential to have immunogenicity. So it actually getting more a lot of intention in this use. And the rationale for using peptide as a delivery tool most commonly is actually through peptide binding to a specific receptor that can go through endocytosis, and which take the peptide and its cargo together into the cell to be delivered inside. So with that, we actually choose a commonly used cell surface endocytosis receptor, and we screen it with our DAO screening platform. And we find a number of peptides that can bind to this receptor successfully. And then to make sure after binding, the peptide actually can get into the cell to deliver the cargo, we used actually to use the flow cytometry. As you can see here on the left side, so from the DAO screen, the platform, we have add a fluorescent dye into the DNA tag of the peptide, and then we incubate it with cell lines either have high expression of the endocytosis receptor or the cell line with low expression level of the receptor. And as you can see clearly, we can show a pretty specific intake of the fluorescent signal in the high expression cell lines compared to the low expression cell line, suggesting this is through a specific receptor-mediated endocytosis process. And then more straightforward, if we're using confocal microscope here on the second row, we can see clearly the fluorescent signal is inside the cell, is in the endosome, indicating that the peptide has successfully been engulfed and getting to the endosome. So in summary of this, those data, we have shown that peptide has a great potential as delivery tool and can be used for conjugating to other modalities to make POC, PDC, and even RDC to deliver those drugs into the body. And then assay methods, including flow cytometry, confocal microscope, can be very helpful for delivery peptide evaluation. And again, those new technologies as DAO and mRNA display platform can also help a lot for drug discovery in this application as well. So that's all I have for our integrated peptide platform. I'm happy to take any questions you guys have. Thank you.
Thank you so much, Dr. Zhongyao Mao and Dr. Yivei Singh, for that excellent presentation. And just a reminder for the audience that you can ask questions through the Q&A box on your screen. And with that, we're going to open it up to our first audience member question. I believe this is for Dr. Mao. This audience member is asking, which screening platform should I choose for my target? Do you have any recommendations?
Yes, thanks for the questions. And I think this is a very, very good question. And we got these kind of questions from our clients and our collaborators a lot. So I would say this is actually a case-by-case question. So as we described before, the DNA-encoded libraries has its unique advantages, mainly by its unnatural amino acids, building blocks, and different cyclization strategies. While mRNA display platform, you can design your own peptide structures pretty easily, and also the library diversity is higher. So based on our previous project experience, we do see for some targets, we didn't really get anything from DNA-encoded libraries, but we do get a very potency hits from the mRNA display. But we also see similar cases vice versa, which means we didn't really get a lot of clustering from the mRNA display, but we do get a lot of potent hits from the DNA-encoded library. So I would say the answer to this question is mainly case by case, but if it's applicable, I would suggest to try both screening platform and also mainly decide what screening platform you will use based on what kind of the target peptide compound you would like to have in the end after the screenings. Or do you want a nature amino acids in your primary hits? What kind of structure of the peptide you want? Thank you.
Okay, thank you very much. Dr. Singh, this audience member is asking for the SPR studies. What is the minimum target purity required for those experiments?
Thank you for the question. So there's not like a clear cut of the purity of the target protein needed for SPR to be performed. However, we would recommend at least 95% of purity to ensure the solid result of the KD we will get.
Okay, thank you. I think we can squeeze in about one more question here. This audience member is asking about membrane protein as a target. We all know that it's kind of like tough to maintain the structure of the membrane protein for affinity-based testing. Do we have any insights, or do you have any insights from WuXi on those kind of targets?
Yeah, maybe I can start first.
Sure,
so membrane protein is really challenging. So based on our previous experience, DNA-encoded library screening is feasible for a lot of membrane proteins. We do get a lot of successful stories and cases. On the other hand, phage display also seems to be really good on the membrane protein selections. For the mRNA displays, we see some successful cases, but we are still accumulating experience. It seems to be challenging for the mRNA display to working on the membrane protein, especially for those nanodisk based. But we are still working on that. Yeah, thank you.
Okay. This audience
I would
member,
like to chime in as well.
Yes.
So
by all means, I'm sorry. Go ahead, Dr. Singh. No, my apologies. Thank you. Go ahead, please.
No, I just want to chime in on the assay part. So for assay perspective, we still recommend like performing those assays mainly in cell-based assay to be able to get more solid data.
Thank you so much. We do have time for one more question. This audience member is asking, does WuXi AppTec have any experience with RDC applications?
Sure, yeah, I will take that one. So we do see a lot of new requests on RDC and a lot of interest in this area. So here in WuXi, we do have comprehensive toolboxes built for RDC application. So we have the capability to characterize like the peptide itself. We have the capability to do the conjugation of the linker, the chelator, the cold metal, hot metal onto the peptide, and we have the capability to look at the whole DMPK of the hot metal as well. So in general, we do have a lot of experience in this field, and if you have any needs, please let us know.
Okay, great. Thank you so much. And that's about the time that we have for today. And this concludes our webinar. Thank you again, Dr. Jungyao Mao and Dr. Yivei Singh, for this excellent and informative presentation, as I mentioned earlier. Any questions that we didn't get to during the webinar will be answered by email. Thank you, participants, for being a great audience, and On24 for the technology and production services. And a big thank you to our sponsor, WuXi AppTec, for making this webinar, this interactive webcast possible. Be sure to check CNN or CNN Online for information on the next edition of CNN webinars. And for CNN webinars, I'm Melissa Romero. Goodbye.